Hello, my name is Gatis Mozoļevskis, and I'm co-founder and CEO of Cellbox Labs. We are the first company in the world that has made advanced gut-on-chip model that is miniature organ replica outside our body that can make a drug development process faster, easier, and more effective. Drug discovery business is expensive. It takes a very long time and takes a lot of money. One of the reasons is that they fail a lot, as much as startups do. And the reason is that in clinical trials, we let through compounds that are toxic to humans. We let through the preclinical stage. And in that part, there is improvement needed. One of the reasons why it happens is because we are using unsuitable test tools. We are testing drugs on poor models. Right now, the golden standards are Petri dishes and animals. Problems with Petri dishes are that their cells are static and they don't experience the same environment as human body do. And animals, although they are living beings, they are still too different, especially their gastrointestinal system. Back in the 80s, Eli Lilly started to sell benoxaprofen. They spent 16 years development, spent a lot of money, expected huge amount of profit. They tested on mice, rabbits, and even monkeys. But what happened is people started dying of this, and it was a complete disaster. So right now, it has been shown that it could have been predicted if they would use organs on chips. We have a solution that combines human organs and Petri dishes, called organs on chips, that is artificial miniature organ outside our body. Let's look into more detail how it works and what it is. It's a plastic chip, less than credit card size. It has small microchannels, less than millimeter in size. And it has porous membrane in between that mimic our organ barrier. What we can do, we can put cells in these channels. For example, we can take gut cells, put in one channel, and blood vessel cells in the other channel. We add flow, as in our bodies, and these cells literally like this environment, and they grow exactly like in our organs. What we can also do, we can put actually microorganisms in it, like bacteria, viruses, and test them. One example is leaky gut. That is a problem that happens when you have your gut barrier integrity is damaged, and all the toxins and bacteria, they get into your blood circulation system. And this can cause a lot of chronic diseases. Regarding gut health, have you ever taken probiotics? Okay, yeah. I see there are a few hands. So there are trillions of microorganisms in our body, and they are called microbiota. They are bacteria, fungi, etc. And these microbiota influences everything in our body. You might have heard that the 21st century has been dubbed century of gut. And this balance can cause diseases like depression, cancer, obesity, and also it plays a very important part in drug efficacy and toxicity. Here is a list of all microbiota test models currently available. As you can see, there's not that many. What we have done, we have taken our chip and we have taken microbiota from a human, and we have put this microbiota inside the chip. The green color, what you see, is actual microscope image of live bacteria. That means live bacteria lives there, and you can do all kinds of experiments and drug tests. One of the topics of this event is space. But what happens when astronauts go to space? There are a lot of physiological challenges, including digestive system. there are companies that specifically develop probiotics for astronauts. But how do you test them? You have to do a space mission. What we do is, in one of the research projects, we simulate that on Earth. We take random positioning machine that turns the enclosure object inside, around, and it falls all the time. And it experiences very little gravity. And we are developing an organ on chip and a protocol that you actually grow gut on chip on Earth on space conditions. The value proposition for organ on chip that is a more accurate in vitro model for faster and more precise decision making in drug discovery process. That consists of a chip and an instrument that runs these chips, and you can run experiments. Who needs that? Big Pharma needs that. There is a study that shows that 87% of compound toxicity could have been predicted if they would use organ on chip. For example, gut on chip alone, it could save $2 billion every year for pharmaceutical companies. But let's imagine if all of us, if everyone, we could make our own personalized organ on chip. Let's meet John. He's in deep tech, and he's busy, and he's building his unicorn. Last night, he went to a conference dinner. Food was very good. He tried everything. But he feels really bad, and he thinks that it might be an allergic reaction. What can he do? He can make his personalized organs and chips from his cells, his own gut, and he can test, can I eat this fish? Can I eat this strawberry on top of creme brulee? Or is this the shrimp cocktail will be the last one? But what you didn't know about John, he's actually an avid smoker. He recently switched to vaping, but that didn't help. And he has diagnosed lung cancer. So what you can do, you actually can take these lung cancer cells, because there are different types of cancers, and then you can put it on a chip. And what you can do is you can test different drugs before you take medicine one by other and trying to find out which one works. So you can see that yellow ones didn't work, green one works, and the red one didn't work. So this is what we are doing as well. We are working with a hospital in Latvia and also Biomedical Research and Study Center to make a proof of study that will show this procedure. Growing these chips, it's not an easy task itself. It has like 100 parameters, like different things that you have to take in mind. But imagine if we want to grow chip for every human being, that means there are different cells for everyone. That gets more complicated. So what we are doing, we are taking microscope images of the chips, we are taking sensor data, then we are working with the Institute of Electronics and Computer Science, and they are using machine learning and artificial intelligence to make an automatic protocol that adjusts to every cells, every person, to make it available. Okay, John is back. He is actually smarter now. He is healthy. And what he has done, he has given stem cells to Clinic. And Clinic has made a bank of his organs, and they are keeping them in freezer. So when he visits the doctor the next time, he tells what his conditions are. Doctor says, okay, you have to take this type of medicine. Come back in 2 days. We will test exactly medicine that will work for you, and it's not going to be with complications. And this is what we envision how personalized medicine will be. We are right now talking about one organ. Imagine if you can combine lung, kidney, heart, brain, gut together. you can make human on a chip. And that gives much more possibilities. You can replace animals in studies. Right now, we kill more than 10 million animals in Europe alone. EU have plans to phase out animal testing by 2050. US Environmental Protection Agency will stop conducting animal studies by 2035. That means all pesticides will be tested some other way than using animals right now. Yeah. And a few words about our company. We are 3 co-founders with background in startups in Silicon Valley, in medical device and microfluidic development, in biology. We have a couple of colleagues developing technology and business. We have collaborators for Latvian Biomedical Research and Studies Center that help to develop biology. If you happen to have a PhD in microfluidics and biology, we are interested to talk with you. And we are raising money to bring this technology to the market. Okay, thank you, and please get in touch. I will stick around. There is a QR code with my LinkedIn. Me and Roberts are here. Thank you.
Thank you so much, Gatis. Thank you.